Assalamu alaikum طلاب, and welcome to part 2 of our lecture that is about the histology of the liver. We discussed in part 1 the functions of the liver. We passed into the vasculature of the liver and how the liver is supplied by two arterial supplies, one from the hepatic artery and one from the portal vein. We discussed how the portal area is formed and the hepatic sinusoids and their function. And now we will continue with the main histological features of the liver. So as we said earlier, the liver is surrounded with a very thin capsule. This capsule sends septa, connective tissue septa, to the inside of the organ, dividing it into loops and lobules. Each liver lobule, as we can see here from the photo, is a hexagonal structure demarcated by connective tissue, and it is formed of mainly central vein in the center of the lobule. As you can see here, central vein, which is the central terminal hepatic venule, which will collect blood from the sinusoidal capillaries of the liver and pass them to the hepatic vein. Radiating from this central vein is an interconnecting plates of hepatocytes. Between them are sinusoidal channels, sinusoidal hepatic channels. So all these red in color are plates of hepatocytes, which are the main type of cells in the liver, and between them is the hepatic sinusoid. In the periphery of each lobule, we can see here it is clearly demarcated by connective tissue, and we have on each angle 6 to 8 portal areas in the periphery of the hepatic lobule. We can see that the portal area is composed of the three important structures that we discussed previously, that are branch of the hepatic artery, branch of portal vein, and a bile duct. And these are called portal trait. Branch of portal vein, branch of hepatic artery, one or two branches of bile conducting system. So what are the main functional cell of the liver? It is called, as we said previously, the hepatocytes. These are the hepatocytes, and they fill the whole lobe and lobule, and they are present on each side of the hepatic sinusoids and surrounding the central vein. If we describe the histological feature of these cells, we can say that these cells are large polygonal cells with centrally located nucleus, eosinophilic cytoplasm due to the presence of large number of mitochondria, and we can say that these cells are highly active. They are very active cells metabolically. They are always busy cells with number of functions: secretion, absorption, synthesis of plasma proteins, synthesis of many other proteins important for the body. So they need large number of mitochondria and large number of other cytoplasmic organelles. That's why they appear eosinophilic in staining. As we can see here, the central vein in the center, surrounded with large number of hepatocytes radiating toward the periphery, and as we said earlier, the hepatocytes surround sinusoidal capillaries. If we look closely at the hepatic lobule, we can see that each hepatocyte, each liver cell or hepatocyte, has three important surfaces. The first surface is called the sinusoidal surface. The first surface called sinusoidal surface, which account for approximately 70% of the total hepatocyte surface. And it's in contact with the hepatic sinusoid, where the hepatocyte is in contact with the blood. These sinusoidal surfaces are covered with short microvilli, as we can see here. The liver cells on the sinusoidal surfaces have short microvilli that protrude into the space of Dizzy. This surface is the surface where materials pass from the blood into the hepatocytes and vice versa from the hepatocytes into the blood. which means this is the surface responsible for absorption of materials from the blood and secretions of materials from the liver cells into the blood as well. The other surface is the canalicular surface, which account for about 15% of the hepatocyte. This is the canalicular surface. As we said, it forms about 15% of the hepatocyte surface, and they are where the hepatocytes are in contact with bile canaliculi, as we can see here. So this, for example, for this hepatocyte, this is the sinusoidal surface, and this is the canalicular surface. Bile canaliculus, this is a bile canaliculus, is formed by the exact opposition of two shallow grooves on the surface of adjacent hepatocytes. So formation of bile canaliculi start near the central vein of the liver, and each bile canaliculus will increase in size as it moves toward the periphery of the lobule. These canaliculi are about 0.5 micrometer in diameter when they initially formed, and they increase in size up to 2.5 micrometer in diameter before they join the bile duct. Hepatocytes, on their canalicular surface, they also have short irregular microvilli, and the cytoplasm of the hepatocytes near the canalicular surface is characterized by the presence of actin filaments. And as you know, actin filaments are filaments that can contract and relax, and that's why we have an idea that this actin filament will influence the rate of flow of bile from the canaliculi into the bile duct at the periphery of the hepatic lobule. In addition, the cytoplasm of the hepatocytes near the canalicular surface contain a large amount of the two most important hepatic enzymes, which are alkaline phosphatase and adenosine triphosphatase, ATP and ALT, and these two enzymes increase where we have cases of liver injury, which mean that they give an indication of abnormal liver function test. The third and the last surface of the hepatocyte is the intercellular surface. And from its name, it's the surface between two adjacent hepatocytes that are not in contact neither with sinusoids nor with bile canaliculi, and it accounts for 15% of the surfaces of the hepatocyte. So in summary, we have three important surfaces for each liver cell. 70% of the liver cells are in contact with the blood sinusoids. 15% of the surface area of each liver cell is in contact with a bile canaliculus, and the other 15% is adjacent to the nearby hepatocyte. Functionally, the scientists have described the histology of the liver and its relation to its function in three ways. The first way is the classic hepatic lobule way of describing the histology of the liver, which we discussed earlier, where the liver is formed of hexagonal lobules. In the center we have a central vein, and in the periphery we have portal areas or portal tracts. In this method of explaining the histology of the liver, we have the central vein in the center, the zone of hepatocytes immediately near the central vein is called the centrilobular area. The middle area is called the mid-zone, and the periphery is called the periportal area. This is the oldest classification of the liver according to its function, and because it's the oldest classification, most of the disease processes is based on this classification. For example, we have a condition known as centrilobular necrosis, where there is death of cells in the centrilobular area of the liver lobule. A second way of looking at the liver in regard to its functional unit is by viewing the portal lobules. This theory of viewing the liver mainly concerned with the bile synthesis and bile drainage from the hepatocytes to the bile duct. So this area is a triangular in shape. The portal lobule is triangular shape lobule with the portal trait in the center and 3 central veins at the angles of the triangle. As we know, the bile always flow from the center of the classic hepatic lobule toward the periphery. And in this viewing of the functional units of the liver, we have the bile flowing from the angles of the triangle toward the center. This concept is more useful when considering the bile secretion function of the liver. The tissue draining the bile into the portal area is roughly triangular in shape, as we said, and the portal area is in the middle. A third way of viewing the liver histology is the theory of hepatic asanas, or liver asanas, as we can see here, where the hepatocytes are stretched between, or the liver asanas, we can say, the liver asanas is a diamond-shaped area stretched between 2 central veins and 2 portal areas at the angle of the diamond. This shape, or this theory, is mainly concentrating on the concept of oxygen supply to the liver cells, where this area here receive the most oxygen supply when compared to other areas of the liver lobule, because they are present, or they are located near the hepatic artery that carry the oxygenated blood into the liver. These hepatocytes that are present in zone 1, this is called zone 1, which is the most oxygenated cells, they are responsible for the oxidative metabolism, such as protein synthesis. However, hepatocytes in zone 3, for example, receive the least oxygenated blood, That's why they suffer the most in cases of ischemic necrosis of the liver. Okay, and their function is mainly related to glycolysis, lipid formation, which are functions that doesn't need oxygen supply as other functions. In the middle we have zone 2, which is intermediate between zone 1 and zone 3 in relation to oxygen supply. The major activities in any given hepatocyte in this area result from the cell adapting to the microenvironment produced by the content of the blood to which it is exposed. These 3 ways of viewing the functional units of the liver, or this architectural arrangement of the liver, remain immutable, or remain unchanged in recent years. This is just simply 3 different ways of looking at the structure of the liver. However, histologically, on a slide, we cannot see a portal lobule. This is an imaginary area. We cannot see a hepatic asanas. Also, this is an imaginary. We only can identify and see the classic hepatic lobule. So by the end of this lecture, we see that we have fulfilled our objectives. We discussed the classic hepatic lobule and how it is formed. We talked about the hepatocytes and their 3 main surfaces: the canalicular, the sinusoidal, and the intercellular surface. We have discussed the portal tracts, hepatic blood sinusoids, space of disease, an important space that we have talked about, the formation of bile canaliculi and the canalicular surface, And lastly, we have discussed the functional units of the liver and its arrangement. See you in the next lecture. Thanks a lot.